Hello, everyone. My name is Mackenzie.
My name is Cole.
And my name is Vanessa.
And welcome to the Couch Potato Lab,
Lab,
the show where we bring the science to you in your home. Now, before we get started, you can download the lab manual at bit.ly/couchpotatolab. That has all the information you'll need in the show, and you can follow along the activity there with us. Now, you can also, throughout the show, ask us your questions. So you can text us in your questions at 306-570-1013. And you can also find us on social media at Eyes Youth. So that's on Instagram, Twitter, Facebook, YouTube, and also on TikTok if you want to see us do some crazy things on there. If you're using Twitter or Facebook, make sure you use that hashtag CouchPotatoLab. You can get a hold of us there. We love hearing from you, hearing your questions, so feel free to send us those questions and what you're doing in on there, and we'll get back to you as soon as we can here on the show. Now, we have a great episode today. We're going to be learning lots of things, so I brought by my pro scientists. So who do I have here to my left?
Well, thank you very much, Mackenzie, for that lovely introduction. My name is Cole. My pronouns are he and him. And a little bit of a fun fact about me is, while I love coming here to work on the Couch Potato Lab, my true passion is actually magic, and I'm a little bit of a magician, so is this your card? Probably was.
Wow. And as I was saying before, my name is Mackenzie. My pronouns are she, her. And a fun fact about myself is that the Eyes team finally inspired me, and I learned my very first TikTok dance yesterday, and I'm very proud of it. Yes.
Nice. My name is Vanessa. My pronouns are she and her. And I also love doing the Couch Potato Lab, but during the day and anytime I'm not at the Couch Potato Lab, I'm actually a pro audience member at magic shows, and it is my card. Thank you, Cole.
Yes.
You did it.
Nice.
Very impressed. All right. So yeah, before we get started, we just want to recognize that we are coming to you live from Treaty 4. So that is home of the Nehewak, Nakaway, Nakota, Lakota, Dakota, as well as the homeland of the Métis-Michif Nation. So we just want to take a minute to recognize that we are coming to you from shared land, and we invite you to recognize where you're situated today, whether that's on another numbered treaty or unceded territory. So please take a second to recognize that before we get started. Cool.
What are you doing there, Mackenzie? We gotta get on with the show. I'm not sure. It's probably not the best time to be sipping on some milk.
I'm just gonna keep going a little.
It's pretty cool how the bubbles start kind of big.
Whoa.
Uh-oh.
Okay, you got some spillage going on. But
oh my goodness.
Vanessa, what I was noticing is that when she started, it was like kind of like one bubble.
Yeah.
But now there's like lots of bubbles.
Mm-hmm.
That's crazy.
It is crazy. So it's actually pretty cool how we start with one bubble, and then suddenly there's pressure on the bubble because Mackenzie keeps on blowing. So that bubble, because it gets too big, it needs to split into two. And then as she keeps blowing, the other bubbles, they get too big, and they then need to split into two, and it keeps on going. So we start off with 1, then we get 2, then we get 4, then we'll get 8, all of that. So it's very interesting how that happens, and yeah, lots of bubbles, big mess now.
That's right. Yeah. So I made so many bubbles. Now, Cole, does that remind you of anything?
It does remind me of something. One of my favorite things, actually, and I think it's actually the topic of today's Couch Potato Lab, and that's called mitosis. Mitosis. Maybe you've never heard that word before, but it's a really, really important process that goes on inside of our bodies. Now, the reason that strawberry milk demonstration reminded me of mitosis is because, Mackenzie, when you first started, it started with one small bubble, but then we grew and grew and grew, and then from 1 we had 2, and then from 2 we had 4 bubbles, and then 8, and then 16, and then we started getting really, really big numbers where I almost lost count. Now, that type of growth is kind of what happens in our cells when they undergo that process that we were talking about earlier called mitosis. Now, Mackenzie, does that process have a special name?
It does. Now, you love mitosis and I love math. And what we call this growth is exponential growth. So I'm going to demonstrate that here. I have a chunk of Play-Doh. So as you can see, I have one ball. Now, in exponential growth, it's going to split into 2. And then what's going to happen next is both of these balls are also going to split into 2. And now these are all going to split into 2. And then we're going to do it again. So they're all going to split into 2. And it keeps on happening. They're going to keep splitting, so they're all going to just keep on splitting in half until we have so many. So as you can see, it started as 1, and now there is so many here. So this is called exponential growth. And I'm actually going to pull out the whiteboard here. And we're going to go into a little bit more detail. It's sometimes a little bit easier if we can see it. So I'm going to pull out our whiteboard here. So very similar to what we were just doing here. We're going to start with 1. Now that 1 is going to split into 2. These are both going to split into 2. And it just keeps going on forever and ever. And they just keep splitting. So if I were to keep going, this would take us the entire episode, all day, all weekend. It just continues on. So instead of just keep on drawing circles, because we know that they constantly are just splitting into 2, we're going to draw a little graph here. So in the first one, we can see we had 1. Then we had 2. Then we had 4. Then we had 1, 2, 3, 4, 5, 6, 7, 8. So what's happening here is this is exponential growth, and we are raising them to the 0. So this is equal to 2 to the 0, 2 to the 1, 2 to the 2, 2 to the 3. Now, if this, you can kind of see the pattern. It has 0, 1, 2, 3. Now, Vanessa, what do you think this exponent number is going to be? Do you have an idea?
If we're going through our pattern, I'd have to say it'd be 4.
That is right. So this will be 4. So we can calculate what 2 to the 4 is, or we could just keep drawing our picture. So we'll go ahead and do that really quick.
That is a lot of circles, Mackenzie.
So many circles. I'm going to run out of room.
Uh-oh.
I might have missed one. Let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So this is 16. So if we were to graph this, we had a lovely graphing episode the other day. It was so much fun. So luckily I watched it and I learned all about graphing. So I'm going to just very quickly draw what a graph would look like. And if you want to learn more about that, tune into that episode. It's very exciting. They did some crazy things on there. It blew my mind. And the graph for this one would look like this. So it would be pretty steady, grow, grow, grow. And then it would just skyrocket. And that is called exponential growth. So it's a little bit slower at the beginning, and then it just skyrockets. And you can see how 1 divided into 16 very, very quickly. And that is how exponential growth works. And that is how our cells are constantly dividing by a process called mitosis. So today, you're in for a treat. We're going to learn lots about mitosis. And luckily, I have my mitosis experts with me, and we're going to be able to go through and learn how the phase works step by step.
That's right. That's right. So mitosis, we go through a lot of different phases, all right? But what's something that's really, really important to know and to understand is that while our cells use mitosis to help us grow, to help us heal, and in some organisms actually to reproduce, they don't spend all of their time in mitosis, okay? So I actually think we have a graphic here that we can show of something called the cell cycle. Now, the cell cycle is showing how the life cycle of a cell kind of plays out. And when we look at that graphic, you're going to see that the majority of the time is spent actually not doing mitosis. Mitosis is only like this little chunk of the time spent in the cell's life. So you can see that M there, the blue chunk of this donut, is when we have mitosis. All those other parts, G1 and S and G2, we don't really need to know exactly what's going on there, but you can kind of think of that as that's when the cell is just carrying out its day-to-day life. It's growing, it might be making some DNA, might be doing some more growing, or whatever that cell is supposed to do. We have lots of different cells, our muscle cells, our bone cells, our nerve cells, our hair cells, and they all have different specialized functions. But you can see that little blue chunk is where we spend just a little bit of time with mitosis. Now, mitosis, there's a lot of different phases, and Vanessa and I are going to show you through all of those phases. So make sure you have the lab manual downloaded and ready to go. And we're going to be making a few different diagrams and demonstrations of the different phases of mitosis.
Perfect. So as we go through this, I invite you to ask us all your questions. So make sure you're texting us. That number again is 306-570-1013. And we also just want to hear from you on social. So you can reach us at Eyes Youth using the hashtag CouchPotatoLab on Twitter, Instagram, Facebook, TikTok, wherever you want. You can find us, ask your questions, and we'll get those answered here on the show. So I believe in the cell cycle, there's many different phases, so we're going to break it down. Now, Vanessa, do you want to start us off today?
Absolutely. So I'm the expert on interphase. This is that part with G1, S, G2, but overall it's interphase. What's going on there is kind of just the cell's not splitting apart. It's just growing. It's getting ready to split apart because lots of the cell's job is to split apart. So in that time, it's going to be growing, getting bigger just in general. It's going to be making more of its little organelles. And the organelles are the little parts of the cell that do special jobs in the cell. So it's making more organelles, that makes it bigger, all of that stuff. And something really important is that it also replicates its DNA. So we have a little picture that I drew for our DNA replication. So we call these little things chromosomes. So right now they are just one chromosome, and the chromosome has everything that your cell needs to do its job, all the information. So that's how it looks at the moment. And then we're going to do our replication. So very fun. We've got an extra one. It comes in, and then it just sticks beside like that. And then in comes our next one, like that. And then in comes our next one, like that. So they're just kind of hanging out like this. And then now we have it replicated. All of our organelles are replicated. The cell is big enough, and it's ready to start splitting apart. That is my expertise. Cole, I think you've got the next phase.
Yes, so interphase is really important, but one thing I want to go back to you on, Vanessa, is the pipe cleaners that we're using to demonstrate as chromosomes. What's actually inside those chromosomes? What's going on that's inside? Because I know you're the expert on interphase. These pipe cleaners represent chromosomes, but what is actually inside chromosomes?
You're so right. I got too ahead of myself. So that's our DNA. We have our DNA give us the information cells need, and strawberries have the same thing, actually. So we've got strawberries right here, and we've got a special solution going on. This is our cellular membrane, which protects the whole cell, and then also the nucleus, which protects the DNA inside of the cell. So that's what we have right here. We crushed up our strawberries in a bag, and then now they look like this with our dish soap. So their cells should be all broken up. And then this is our alcohol. It actually takes the DNA out and will make a layer of DNA for us. So we're just going to pour in. You just kind of eyeball it. We'll just pour some DNA in, pour some alcohol in. And then it should pull all of the DNA up. Okay, so the reason that it is sitting on top of the dish soap and strawberry mixture is because it's a different density. So the alcohol the isopropyl is lighter than the strawberries and dish soap, so it's able to sit right on top. I see a little bit coming along, so you just gotta be really gentle. And I'm just gonna try and pull as much out as I can. I think I've got a bit. And there's a little bit of strawberry in there too, because the chunks. But you can see, I'll put my hand behind it, maybe I'll make it better. But if you can see that slimy thing, that's strawberry DNA right there. So that is like the chromosomes and all that stuff just in a slime for us right there.
Day 2, I brought in these scientists so I could be an expert on the cell cycle by the end of this episode 2. So as they're going through each phase, I'm going to go ahead and jot some notes down here on the whiteboard here, and then we can refer back to the cycle at the end. And if I make any mistakes or I have anything wrong, my scientists will be sure to correct me. So they're going to be keeping an eye on it, so will I, and then we'll share it with you as we go too. So Vanessa just went through the interphase, so I'm going to go ahead here, and I'm going to draw my cell. And Vanessa was saying how there's DNA inside the nucleus. So I'm going to draw a quick little nucleus here. That's going to be that. And then the DNA that she was just talking about is inside that nucleus. So I'm just going to draw some squiggly lines here inside, and that's going to be my DNA for now. And I'm going to just label that interphase. What do you think, scientists? Did I do a good job there?
That looks great.
That looks fantastic. Really,
right.
really good. Yeah.
So I'm good on interphase. Let's move it on over to Cole, and we'll learn about our next phase in the cell cycle.
Excellent. Thank you so much, Mackenzie. The next phase in the cell cycle is called prophase. Prophase. Now, we learned from Vanessa in interphase, one thing that happens with our chromosomes, our DNA, is that it replicates. Now, normally, our cells, when we think back to that cell cycle and we spend most of the time not in mitosis, when our cells aren't in mitosis, they have 46 chromosomes normally. All right? After we duplicate everything, we make a copy of everything, now we have 92 chromosomes in our cell. There's a lot going on. So to help make things a little bit more clear, what's going to happen, and Vanessa sort of showed this, is the chromosomes are going to start to pair up. So what you can do is the chromosomes are going to find their sort of duplicated pair, their partner, if you will. And you can take your similarly colored pipe cleaners, and all you need to do is take them and sort of wind one around. And we're going to make these cool, funky little X shapes, all right? Sort of like this, okay? So what we did here is all of these chromosomes are now finding their partners, okay? So I've got a purple, I've got a red, and I have an orange. Another important thing that happens in prophase, if we look at this diagram right here, is that in prophase, if you think back to interphase and Nessa's drawing, she had 2 circles. I only have 1 circle. The reason that is because that inner circle is called the nucleus, and it actually disappears during prophase. So the nucleus is appearing, and the reason that has to happen is because the nucleus is sort of the protective barrier that keeps all of those chromosomes safe inside the cell. If we want to duplicate them, if we want to split them and make them into new cells, you get rid of that nucleus first. So the nucleus is gone, and another thing that happens are these two kind of look like spiders in the drawing, but these are spindle fibers, sort of long, kind of stringy looking things that are coming out. Those spindle fibers are coming out, and we're going to see later on a different phase, I don't want to spoil it, what those are exactly used for, but you can take your paired chromosomes now, and you can sort of just stick them on. We're using tape just so we can better show you folks at home. But what we've got is our chromosomes paired up, and they're kind of just floating around the cell, okay? They are not really in any specific order. They might get ordered later when we learn about some other phases, but right now they're sort of just hanging out. The important thing to know is that we have all of the pairs there, right? So those chromosomes that have duplicated have now found their partner, and those pairs are, the way I like to remember prophase is for P pairing up, or